Thanks for being here, Herb.
Yeah, thank you so much. I appreciate this chance to do this. I want to give a little bit of background about where I am right now. I spent 38 years in the ag chem industry, decided to retire and grow citrus. And in doing so, I discovered regenerative and organic, and what a exciting way to grow and to work on the soil particularly. So I planted a grove. I've got it's small citrus wise. It's 1,300 trees. I'm packed onto 5 acres, and these are some of the goals that I have growing regeneratively. Ultimately, it's to have a more healthy tree based on the health of the soil. High bricks in the tree and also in the fruit, tight internodes, which you kind of see on some of these pictures, a dense tree. Ultimately, that could lead, hopefully, to low insect pressure. Something that's important for citrus growers, we strive for as many flushes of leaves as we can. We get the entire new flush of leaf on the tree. We can get up to 5 flushes a year. So that's my goal. If the trees are ultimately healthy, I'd have that higher level of growth. So to grow regeneratively, I feed the microbes constantly, either with root exudates from these cover crops, or just literally through the irrigation. I send a lot of food to the microbes. And the ultimate desire commercially is to end up with nutrient-dense fruit. So one of the things that I do is grow cover crops. And this is how nice and lush they end up getting in the wintertime. And it's clovers, hairy vetch, Austrian winter pea, ryegrass, black oats, daikon radish, kale, turnips. And then I mow it to the row always. So I'm trying to provide nutrients back to the tree, let my cover crops provide nutrients, and then really provide habitat for microbes, and keep the temperature down, help hold in moisture. So I plant 10 different species of cover crops in the fall, and then a different 10 in the spring. So one of the satisfactions that I've gotten, or proof that things are working, is with a penetrometer. You just put 200 pounds of pressure on it, and in the middle, it penetrates about 3 inches. So that's the natural compaction of the soil that we have. And when I go between the trees in the row, the penetrometer, it goes at least 2 feet in. A lot of times it just sinks easily all the way to the handles. It's pretty amazing. So it's a combination. This is roots from the cover crops, roots from the trees, but really it's the microbes are adding a lot porosity in the soil. So hopefully that's going to really help trees as they grow. This picture was taken just after the terrible freeze that we experienced a little over a year ago. So everything is in tough shape. This is one of the results that I've started to see. My trees are just 3 years old. One of the first fruits that I got were page mandarins. And on the right are the page mandarins out of my grove. And then I went to a commercial grove about 15 miles away that had page mandarins, and picked them. And there's this dramatic difference in color, flavor, aroma, and everything. So my hope is I've juiced both mandarins and a grapefruit variety from a different conventional farm, and hope to send them to a lab for nutrient analysis so that we can actually have some documentation of what regenerative organic practices are doing. So I want to shift, and that's enough about me. we're going to look at EnSoil Algae. I've done two sets of trials. One was on my own grove on citrus, some real interesting results. And then another was on a large commercial vegetable farm, a trial on peppers. But this is a discovery of Dr. James White at Rutgers. And this is just quite incredible. And it revealed a lot about EnSoil. EnSoil is Chlorella vulgaris, which is an algae species. What Dr. White found out is that not only does the EnSoil, the chlorella, go into the root as an endophyte, but it is full of bacteria, so it carries them into the root. So it's a carrier, and then it also has its own endophytes, which it releases into the root. So this is an exciting discovery, and if you've ever watched Dr. White's work, it's incredible, this definition of the rhizophagy cycle, which is purely the interaction of endophytic bacteria, which are living in all of our plant roots, and they actually stimulate root hair development. So the trials that I've done have just been a small step to try to elucidate a little bit what the effect of EnSoil and its bacteria that it carries on the microbiome, and then ultimately on the plant, the crop plant. So this is the trial that I did on my own farm. I put everything I do is, because I came from research. I did 25 years of field testing with a couple of different chemical companies. And so obviously everything we did was replicated, and that added validity, we felt like, to everything we did. So on my own farm, it's really turned into a research farm. I have about 10 different varieties, but most of the blocks I've got them flagged off into replicated trials. So I put out this trial with EnSoil Algae, and I put it on 2 different varieties, which was very interesting. I used page mandarins and honeybells. And I got different responses with the 2 varieties. So they were at probably different physiological stages. So when there was a soil impact, they responded differently. But this was the effect of EnSoil. The one on the left is a soil treatment. And so what we were actually testing was, a soil treatment or a foliar treatment going to be better with EnSoil? And the foliar treatment, although there was some soil responses, there wasn't a visual response above ground in the tree. So this is what I saw. So it took the most important thing, like I mentioned, to me was leaf flush. And so I, from the timing of application, I measured leaf flush at 2.5 weeks after application, 5 weeks, and 8 weeks. And this is what happened. These are the results on the page mandarin. So the 3 graphs on the left are the 3 timings, and those are the soil applications. So I was at over 50% leaf flush at 2.5 weeks. And then obviously the flush happens and then it declines. So the foliar spray was, just barely visible, 20%, just the same as the untreated. So the foliar spray. And so their initiation of leaf flush was really after 8 weeks. So essentially what happened with EnSoil is we moved leaf flush 5 weeks earlier than it would have been, which is quite dramatic and something that's real significant in what I'm trying to do to pack in those 5 leaf flush during the year. So the question is, why did it do that? So in addition to the above ground evaluations, the numbers that I took, we did quite a few tests below ground. We did a Haney soil health test on each of the 3 treatments. untreated foliar and soil. And then we did a biomakers BeCrop. And if you're not familiar, I think I'll take a minute and just and jump over to the BeCrop. And I'm actually going to show you results of my own farm. Let me know, Camille, if this went over.
You're good. We
so
got it.
This is what a BeCrop test looks like. So it's basically it's a window into the soil. Every circle in this diagram is a separate species. And so it says up at the top, this is my regenerative treatment after, this is on my trees in a different trial. I'm just using this to explain and to visualize what BeCrop does. I have a large replicated trial, 135 trees, that's conventional, regenerative, and then a third treatment that's indigenous. So it's not adding any foreign microbes, just waiting on indigenous. So the indigenous and the regenerative are both completely organic. And then the conventional is Roundup, bare soil, granular fertilizer, synthetic pesticides, just what a traditional grower would do. So this is what I love in the regenerative. What you see here is diversity. Their results are really interesting. You can click on any of these groups at the right, and it pulls up just those species. Archaea, these are the extremophiles bacteria. Every one of them are nitrogen fixers. So that's neat. The bacteria, there's a lot of diversity. So total, there's 1,064 species were detected in my regenerative sample. That's the different bacteria. The nice thing is there's a lot of diversity, and they're in relatively small quantities. There's just a few ectomycorrhizal fungi, no endos, or just one, and a few yeast and the fungi. So this is great. And obviously, anybody with trees, the goal is to get a high fungal to bacterial ratio. Ultimately, we'd like it to be 5-to-1 or better. But you can see from the total, I think I've reached 1-to-1, and maybe it's a little bit better. In total quantity, the size, each circle is a different species. Here's the largest fungi is Curvularia. It can be a leaf spot pathogen, especially to corn, but mostly it's a decay fungicide. I generally look at this, and then I go through and find there's penicillium, which is great. Pyrenocotopsis, 2 of those species, and an aspergillus, which can be a pathogen. But the diversity is great. And I'm going to switch over and show you the conventional. This is the conventional treatment. Roundup, bare ground, completely different situation. One fungal species, Lycoperdon, has dominated and taken over. It's 89% of all the fungal species that are detected. Lycoperdon is the dwarf puffball. Absolutely worthless. I assume that it's driven out most of the other fungal species. If you look, there's not much of them left, and they're in extremely small quantities. So basically, with the bare ground and synthetic treatments, which there have been a couple of fungicide treatments applied to the tree and another one to the soil, but the bare ground basically has caused this conversion. The field started more like what the regenerative looked like, a decent amount of diversity. Up at the top, it says there's 836 species there. So 200 less species than in my regenerative. But incredible visual of what's going on in soil when it's farmed conventionally. And then the other thing that's so evident here is the bacterial to fungal ratio. So totally dominated by bacteria. The archaea, about the same amount. So they are holding fast, but a lot of bacteria species. So it looks to me like we've doubled or more the bacterial species. So that's there's a lot of ways to look at. This is a dashboard of a BeCrop sample sent to biomakers. So I've been able to follow this test of mine for 3 years. So it's fascinating to look at it and visually almost have a window into the soil and see what's going on. So I'm showing this to show that we have taken those same BeCrop samples in this trial and in the pepper trial that I'm gonna show also. So what was going on in the soil? This shows that there was a little bit of an increase in the number of species, probably not significant. But one of the things that biomakers does is they categorize all of those species that they find, and they know the effectiveness of each one, what it does, if it's a nitrogen fixer or if it opens up phosphorus pathways or potassium. There's actually 40 different categories that they place all of the microbes into, and then they give kind of a summary of what that community of microbes would do for each of those hormonal and growth responses and anti-biological effects, bactericidal, fungicidal. So both of the treatments, foliar and soil, had an impact on nitrogen pathway, phosphorus and potassium pathway. The soil one increased biodiversity, which there's a fascinating component to it. So the Haney soil health test actually proved that biodiversity. There's a measure of how much microbial activity is going on. But the most fascinating thing I found from that, the soil out of the treated that had the EnSoil on it, they measure, the Haney measures both nitrate nitrogen and ammoniacal or ammonia nitrogen, form of nitrogen. And there was really not much change in the untreated in the foliar treatment. But in the foliar, the soil applied EnSoil, the nitrate completely disappeared. So to me, I mean, we know the cycle that the microbes go through. They convert nitrate to ammonia, ammonia to peptides, peptides to amino acids, and then finally to proteins. So the nitrate disappeared. So the microbial activity was consuming nitrate. Almost all of the ammonia disappeared. So it showed, when the EnSoil was treated, that test showed basically no nitrogen left in the soil. It had all been converted to amino acids and proteins. That was an exciting validation. Both of these things validated what I saw above ground, which was that leaf flush response. This is another one of the way that EnSoil, the BeCrop test, shows visually what's going on. Carbon and then NPK. The blue is a higher value than the yellows, and the dark yellow is the least amount. So both the EnSoil treatments showed a numerical response there. And then I looked at the literally, this is called Explorer in BeCrop, and saw these. So here's some of the results that have indicators of what's going on. So you see, I picked the largest fungi or the largest bacteria was Sphingomonas, which is basically a decay bacteria, and it declined. It was 12% in the untreated, and it had dropped down to 5% when EnSoil was applied to the soil. And then you see the fungal presence. Some of the fungal species increased when the EnSoil was used. So probably a lot of things going on in that soil that it'll take time to discern all those things. So in BioMakers, you can actually use a different format and compare 3 treatments at a time. So I picked out species that had high levels and put them into this format. And the untreated is the one on top. And then the foliar is the purple, and the reddish is the irrigated. So it was the soil applied. So looking at different species, alternaria with the soil applied was greatly reduced. Interesting. Streptomyces, which we all know is a beneficial, really dramatic increase, tripling of in the soil applied EnSoil. Mycobacterium, which is a nitrogen fixer, and it does decomposition. And this one jumped up 3-4x with the soil application. All of these are indicators that there's a lot going on. And then this one is the real mystery. We always think of Fusarium as a pathogen, but there has been tremendous new work showing that several of these pathogens, and Fusarium is the one that's been used as a test case over and over, until it detects a weak plant, it's a beneficial in the soil. So my first glance, I thought, oh my goodness, we've doubled, quadrupled the amount of Fusarium. But if the trees are healthy, then the Fusarium is actually acting as a beneficial. So that's the results of the trial that I did. The exciting thing was to see that EnSoil, when it was applied to the soil, caused some real significant activity and the tree responded by a real change in growth pattern. Now, as I mentioned before, the change was really in the page mandarins, and I didn't see a visual change in the Honeybell variety, although those samples were pulled from both varieties mixed. So the same activity was going on in the soil, but in that case, the Honeybell didn't respond to that in the same way that the other variety did. So let me shift gears a little bit, and we're going to have plenty of times for questions. But now I want to go to this commercial pepper trial that I did with Enlightened Soil. It was in Cool Springs, Georgia. Southern Valley is a large commercial vegetable farm. To set it up a little bit, we actually did this time. The blocks are so large, the field was 10-acre blocks. So we did replicates, 3 replicates within each of the 10-acre blocks. And we tried to match as close as possible 2 blocks that were adjacent to each other. They actually almost wrapped around each other. And this was the timing of the applications. So we did the injection with the grower's help into their irrigation system. So we picked one of the 10-acre blocks and injected EnSoil Algae. And the grower was applying a significant amount of biology himself. So he did quite a variety of different things. There were some commercial inoculants and several that he fermented himself. And he had 7 times during the growing season, he actually added biology to the field. So there was a lot of activity already there. And we chose to do our applications 2 at the front stage. So this is the first application timing. Probably it's just a little more than 2 weeks after the transplants went in the ground. And then we did a second application 14 days later. So the thought was get the transplants a really good start with the help of EnSoil. And then we waited until harvest began to do the final and third application, hoping to give the plant a boost late in the season. So it was actually 7 weeks between the first 2 applications and the final application. But the final application, there were 7 harvests, and there were actually 9 harvests, and the last application was synchronized with the second or third harvest timing, depending on the field. So here's a diagram of the field. Block A, 9.5 acres, was the treated. And then block B, which is adjacent to on one side, and then it's on the other side of a pond. That dark area in between A and B is the irrigation pond, which is going to end up turning out to be very significant in this. So all the irrigation water comes from that pond, and anything that's injected for fertility or biology or the EnSoil comes from water in the pond. So this time, BeCrop, because it was a replicated sampling, they did an incredible analysis of those 40 categories that they group everything into. And these were the categories that came out with statistically significant differences. And it's pretty exciting. There's 12 real changes that we saw out of those different categories. If you just kind of look down that list, nitrogen release, phosphorus assimilation, iron assimilation, 7%, sulfur, gibberellic acid production, 40% increase. This is just the EnSoil compared to the untreated. 40%. Heavy metal resistance, 70% increase in that activity, highly significant. Abscisic acid, which has a whole list of hormonal effects, and siderophores, which I actually had to look up and remind myself they do iron chelation, they do some biocontrol, and then they have quite a list of PGR, plant growth regulator activity. So a 38% in siderophores. And then, as you can imagine, because this population changes with the EnSoil, which it's bringing in bacteria inside the algae. So we've got this absolute explosion in the bactericidal agents that are detected. 21,000%. And fungicidal agents went up 280%, so tripling. Nematicidal agents doubled. And then gray mold was reduced 74%. But let's kind of glance through some of the specifics, pull out some of these specific microbes. Globosporangium is actually what we used to call Pythium. And so on the top are the 3 treatments, which 3 reps out of each side. So six pulls. A lot of variability in this Pythium pathogen, which is a real, it's so similar to Phytophthora, and this farm has a battle continually, especially in several of these crops with Phytophthora capsicii. Pythium is more of a seedling disease, and we put them in as transplants. So you can see between the pre-sample and then just 7 days later, big drops, and the drops occurred both in the EnSoil and the untreated, but interesting to see the dynamics of what's going on in the field. Didymiella is actually a pepper disease. It's a stem rot. Same thing, very interesting between the pre-sample and then the two sets of samples later on. A big drop in Didymiella, but you can see there is one sample, there was a hot spot in the field detected way out on the right. 54% of all the fungi were Didymiella in just that one sample. So you also see the variability within the field. On this one, you can see the light blue circles are actually the Pythium. So several different Pythium species that are there in this field. High levels in all the samples. This Phingomonas, which is beneficial, it has some phytohormonal effects, and it remediates environmental contaminants. Really interesting. Hadn't heard of it before until I started pulling these BeCrop samples. So really nice increase as the season goes on. You see these big jumps. And then this one absolutely threw me for a loop, Alternaria ecornii. And this really concerned me when I got the samples back and could not figure why we had this incredible jump. And I'm going to show you what it looked like in the samples. I pulled out two of the untreated and then two of the EnSoil. This is an Alternaria species. But finally, I looked it up. It's actually a pathogen of water hyacinth. And we are irrigating out of a pond which had water hyacinths around the edge. So what a story to realize that pond water is bringing its own pathogen. Now, it probably doesn't carry over into any above ground plant that we know of, but how dominant it became just by irrigation out of this pond water and the amount of inoculum load that it's bringing in and probably altering a lot of the species, the balances that are there going on in the soil. But what's the ultimate good effect? Before we go into questions, I'll just say that the EnSoil treatment, that 10-acre block, had a 13% yield increase. Doesn't sound huge, but so yield was taken. This grower's meticulous, counts every box of fruit that comes out. 45,000 pounds in the untreated and 51,000 pounds of peppers. These are bell peppers. In the EnSoil treated block, it's a 6,000 pound increase in the EnSoil. Now, I checked on the price. The average price of pepper during that season was 68 cents a pound. So a 6,000 pound increase translates to a big financial effect. The goal when we do it in the future, there were other blocks of peppers nearby, and there is some variability when doing trials, yield variability just from these great big blocks. But our goal is to try to do the trial again, either in this crop or a different crop on their farm, to find an area where there's more blocks and actually use the blocks as replication. And that would add a lot more validity to the yield response. So I'd like to, there's some things obviously which can hurt. I think that probably Roundup strips for bare ground is a detriment. Some of the, I mean, every step in organic or regenerative is probably helping the microbial community, the population down there. But I think cover crops are a real significant one. Even if they're out in the row, we know the citrus roots are going all the way out there. So that's a contribution to the soil health and the citrus health. My hope is if I can see some differences. I mean, since I am HLB free, I'm sort of almost like a test tube environment, that if I can find and show some things that maybe it'll have some pertinence just to general tree health, that it would be harder to test in your HLB environment down there. So maybe we can determine some things. My goal is that I see a significant difference in fruit. Next year, when I get fruit off of that conventional versus regenerative trial, I hope to see that same dramatic difference that I saw go into a commercial grove outside, to look up and remind myself that they do iron chelation, they do some biocontrol, and then they have quite a list of PGR, plant growth regulator activity. So a 38% insideriforce. And then, as you can imagine, because this population changes with the EnSoil, which it's bringing in bacteria inside the algae. So we've got this absolute explosion in the bactericidal agents that are detected, 21,000%. And fungicidal agents went up 280%, so tripling. Nematicidal agents doubled. And then gray mold was reduced 74%. But let's kind of glance through some of the specifics, pull out some of these specific microbes. Globosporangium is actually what we used to call pythium. And so on the top are the pre-treatments, which 3 reps out of each side. So six pulls, a lot of variability in this pythium pathogen, which is a real, it's so similar to Phytophthora, and this farm has a battle continually, especially in several of these crops, with Phytophthora capsicii. Pythium is more of a seedling disease, and we put them in as transplants. So you can see between the pre-sample and then just 7 days later, big drops. And the drops occurred both in the EnSoil and the untreated, but interesting to see the dynamics of what's going on in the field. Didyomela is actually a pepper disease. It's a stem rot. Same thing, very interesting between the pre-sample and then the 2 sets of samples later on. A big drop in Didyomela, but you can see there is one sample, there was a hot spot in the field detected way out on the right. 54% of all the fungi were Didyomela in just that one sample. So you also see the variability within the field. On this one, you can see the light blue circles are actually the pythium. So several different pythium species that are there in this field, high levels in all the samples. This Phingomonas, which is beneficial, it has some phytohormonal effects, and it remediates environmental contaminants. Really interesting. Hadn't heard of it before until I started pulling these BeCrop samples. So really nice increase as the season goes on. You see these big jumps. And then this one absolutely threw me for a loop, Alternaria ecornii. And this really concerned me when I got the samples back, and could not figure why we had this incredible jump. And I'm going to show you what it looked like in the samples. I pulled out 2 of the untreated and then 2 of the EnSoil. This is an Alternaria species. But finally, I looked it up. It's actually a pathogen of water hyacinth. And we are irrigating out of a pond which had water hyacinths around the edge. So what a story to realize that pond water is bringing its own pathogen. Now, it probably doesn't carry over into any above ground plant that we know of, but how dominant it became just by irrigation out of this pond water and the amount of inoculum load that it's bringing in, and probably altering a lot of the species, the balances that are there going on in the soil. But what's the ultimate good effect? Before we go into questions, I'll just say that the EnSoil treatment, that block, that 10-acre block, had a 13% yield increase. Doesn't sound huge, but so yield was taken. This grower's meticulous, counts every box of fruit that comes out. 45,000 pounds in the untreated and 51,000 pounds of peppers. These are bell peppers in the EnSoil treated block. It's a 6,000 pound increase in the EnSoil. Now, I checked on the price. The average price of pepper during that season was 68 cents a pound. So a 6,000 pound increase translates to a big financial effect. The goal when we do it in the future, there were other blocks of peppers nearby, and there is some variability when doing trials, yield variability just from these great big blocks. But our goal is to try to do the trial again, either in this crop or a different crop on their farm, to find an area where there's more blocks and actually use the blocks as replication. And that would add a lot more validity to the yield response. So I'd like to turn it over now. Hey, Meredith.
How are you?
Great.
Yeah, I agree with Camille. I've read and poured through all of these reports, and we've gone over them so many times that it's a proud moment to hear them projected and reviewed here and shared. Yeah, I would love, I don't know what everyone's experience here is on the call, but I loved that when we learned that about Fusarium, and if there's anything that you can, elaborate on, overabundance of some species and how they affect plants. And I know one thing we've seen with the bee crops in the past is this balancing of fungal to bacterial ratios, per crop. I've kind of been explaining that as crop needs based on crop needs. So could you talk about that, where you've seen that elsewhere?
Was that to me or to anybody?
That's to you, Herb. Or that's to you, Herb, or if anybody else has anything to add to that conversation.
It's almost like it's new science, the things that we're learning about these, how a pathogen is not, it's not that simple. It's not just a pathogen. But in hearing you say that, it actually makes me think those slides I showed of my own field, regenerative versus conventional, that regenerative has gotten EnSoil in the last 2 years over and over again. So I've used it probably 3 or 4 times a year. And then we saw that beautiful increase in fungal population. And so it's now, I think, outpacing bacterial, which is the direction I want to go. So yeah, EnSoil could certainly be playing a big part in that. That's an interesting idea. I hadn't thought about it exactly that way. Be interested in anybody else's experience or things they've heard about this new way of looking at pathogens.
This Jack, I have a question on your comment you just had. You said you've been using EnSoil the algae for 2 or 3 years. Have you seen an increase in your fungal bacterial ratio as you progress through the years?
It's been dramatic.
Herb, you're having some connection issues.
In my 45 trees that are just conventional. And so they've never gotten in soil. I just, pardon? Are we locked? Is it?
Yeah, we can hear you now. Would you mind repeating what you just said? I think a lot of us missed it.
Yeah. So because I do have an untreated that's never, it's never gotten any biology, it's just been treated completely conventionally. And it's 3 reps, 15 trees in a row. So it's 45 trees. And although, I mean, it's frustrating, the trees are growing well, but they're in that almost sterile environment that's being dominated by that single fungal species that's a puffball. Just completely of no use. I've lost all the diversity of fungal species. So now it's gotten other inoculants, mycorrhizae, which I don't think took a lot of bacterial inoculants, and it's just gotten, the microbes have been fed. So, but EnSoil was one of the big contributors, something that I used regularly in that block. And I think it was a big transformation.
But I guess I missed it maybe while you were locked up. Over the years, did you see a linear increase, or how did it change over the time period?
Yes, it changed dramatically. So the fungal ratio went down significantly, especially in this last year, which was the third production year. The fungal ratio dropped way off, and it's increasing steadily in the regenerative treatments, which include EnSoil as one of the major inoculants that I use.
Thanks.
Hey, Herb,
Yes.
this Thor Hatton down in South Florida, we spoke on a couple other biomakers webinars, and
Sure. Yes.
I think you shared with me that you're not infected with HLB yet, are you?
We are not, fortunately. Yeah. It's not made its way here. I'm afraid it will.
Well, probably unfortunately, but I wanted to ask you a question. Do you see a marriage between what we want to call regenerative or using biology in unison with our conventional means of, cultural practices on citrus?
I think definitely. So there's some things obviously which can hurt. I think that probably Roundup strips for bare ground is a detriment. Some of I mean, every step in organic or regenerative is probably helping the microbial community, the population down there. But I think cover crops are a real significant one. Even if they're out in the row, we know the citrus roots are going all the way out there. So that's a contribution to the soil health and the citrus health. My hope is if I can see some differences, I mean, since I am HLB free, I'm sort of almost like a test tube environment, that if I can find and show some things that maybe it'll have some pertinence just to general tree health, that it would be harder to test in your HLB environment down there. So maybe we can determine some things. My goal is that I see a significant difference in fruit. Next year, when I get fruit off of that conventional versus regenerative trial, I hope to see that same dramatic difference that I saw go into a commercial grove outside.
Yeah.
Unfortunately, I did the trial on Shiranuis, which is a slow growing tree. So hopefully I'll have fruit next year.
I'll share this. I just finished up a trial in DeSoto County on the Valencia block, and we didn't use any algae, but we added biology, and we pulled Haney and BeCrop tests prior to applicating. And here's what I saw, and this is where I think an algae product probably has a fit, is that we went out with 16 ounces in the microjet irrigations. And we didn't see a whole lot after the 30 days post that first application, but after the second application, that's when we started cranking up the biology in that soil.
Mhm.
And I think the aspect of, the algae in there could probably help fuel and feed the biology a little bit, without a doubt. And so we saw those bacterial fungal ratios improve. And what was really exciting to me is we popped a flush on those trees.
Yeah.
We kind of wake the hormonal response up on those trees, and they popped some flush out, and they really started blooming prior to our conventional time of, for these valencias down here in South Florida to bloom. But just for the group, that the tool that I see in utilizing the BeCrop test is, you're just not going to see data like this in a conventional soil test. But after that second application, we opened up those potassium pathways, and you and I both know we need cations to make bricks and sugar, to try to get back to that 7-pound orange, and we want to, I hope we're going to see the same thing on sugarcane, which we just started. So I don't know if you wanted to comment on that.
I think it's fantastic what you saw, that your trees flushed. That's exactly the same response I saw on one of my varieties. And I mean, we know there's a lot going on inside the tree to make it do that, and to be able to put it on the soil and bring that about is fantastic. So
last comment, and in summary, I'll close. But very well that large commercial citrus blocks in central South Florida, whether it's on the ridge or in the flatwoods, that we've glyphosated the heck out of underneath that canopy. So there's like no biological life there at all.
Yeah.
And so, but not only to point fingers at the citrus industry and the use of those herbicides, we see the same thing. My family, we grow sugarcane, and we're talking about black muck soils with 44% organic matter, high CECs, but there's still no biology in those black soils on the south end of the lake. So it's going to be some exciting work that we're going to probably look at here real quick.
I think one of the things that could be done in both those situations is use some of these inoculants. I use some bacterial inoculants. I use a mycorrhizal inoculant. And then I think the EnSoil stimulates both of those. This year I'm going to a product that almost looks like a little honeycomb that does the same thing as biochar, that serves as a habitat for the microbes, and it's got nutrients in with it. So yeah, if after all that herbicide, and it's a more sterile soil environment, and so bringing it back, cover crops would help with that. But inoculants and then trying to feed the microbes, which I think is probably what EnSoil is hoping to do. You saw a difference, and I think that's really significant.
Herb, I would love to know what the product is, the honeycomb, inoculated honeycomb. Sounds
I'm gonna
really interesting.
just sort of discovered it today, and ordered some product.
Darcy said that they're trialing some wood vinegar this year, and they'll have to compare with you, Herb.
That's really interesting. I have some, and I'm not exactly sure how to use it yet, the wood vinegar, but that would be interesting. I'm looking for this product as we talk, but any other questions?